Hey guys, this is a super brief introduction to the normal flora exercise. I just wanted to share a whiteboard with you. This is what you would have seen had you come into class today. This is the normal flora exercise. Each person would have been responsible for streaking, for swabbing 3 different areas of the body, the nasal cavity or skin, the throat, and the rectum, and then streaking these out onto selective and/or differential media to isolate out individual organisms, and then with a gram stain and a couple of other tests to come up with a genus and species name of a few different of your isolates. So let me just briefly review each of these different body sites and how you would have gone about this physical lab. For the nasal cavity, this would be a sterile swab dipped in sterile water and then swabbing just inside the nostril, just one of them. Some people choose to do right outside the nostril or even just behind the ear. These are all nice hot spots for skin-based microorganisms. For the technique behind the streaking here, you would have taken that swab, and in quadrant 1 with that rolling swab technique, you inoculate that first quadrant. Switch to your inoculating loop. For the rest of these 3 would be loop. And this first one would be swab. Right, and then proceed with a streak, with a streak for isolation here. Resterilize, resterilize. And you are looking for individual colonies somewhere out here or out here. So for the nasal cavity, we use MSA. This one stands for, if you can recall, M is for mannitol, S is for salt, and A is for agar. If you recall, these have 2 key ingredients, salt and mannitol. The high salt concentration makes this selective for halophiles, which becomes important for this isolation. We are trying to make sure that we eliminate transient bacteria, right? The ones that are there in passing, we breathed them in, we rubbed our hands on a surface and touched them to our nose, something like that. Most of those are not halophilic bacteria, but our skin organisms, because our skin is very salty in the first place, these guys are adapted to that high salt concentration, and so we select for just those resident flora with our mannitol salt agar. This also allows us, because of the mannitol, to differentiate organisms based on mannitol fermentation. And this will allow you to really distinguish between some of the really common skin organisms, in particular Staphylococcus aureus, that many of us carry as normal flora. They are mannitol fermentation positive versus Staphylococcus epidermidis and are negative for mannitol fermentation. You also might see Micrococcus luteus on here, another really common skin organism. They're also positive for mannitol fermentation, but those guys can be differentiated based on a gram stain. So you would do a gram stain here to visualize each of these isolates here, and then differentiate them based on both gram distinction and mannitol fermentation capacity. In the Canvas module on our pages, I have a whole bunch of resources for you here. You have a results page that shows you mannitol salt agar streaked with a couple different types of skin organisms, and your job is to identify them based on the gram stain and how they look on that mannitol salt agar to give the genus and species level identification to these common organisms. For the throat, this was a swab at the tonsil area, so we're trying to avoid the oral cavity and just get back in the throat. Really common organisms of the throat are Streptococci, and in fact the Streptococcus genus name, a whole bunch of these guys are in the throat. They all have different levels of hemolytic capacity, so we use blood agar to differentiate them based on hemolysis. If you recall, this is alpha, beta, and gamma. Alpha is partial, and gamma or beta is complete hemolysis, and gamma is nothing. So you'll use this blood agar, and again, on our results page, I have an example of a really great streak on blood agar with a whole bunch of different types of bacteria. And I've circled an alpha, beta, and gamma bacteria and identified them based on, or shown you their gram characteristics and their catalase test. And with these combinations, hemolysis, gram distinction, and catalase, you can identify genus and species of many of these really common throat organisms. And there's some good discussion questions for you there. And then finally, the rectal area. This is a swab about 1/4 of an inch into the rectum. You should be grateful that you did not have to participate in this, but it is a nice bonding activity for those in lab. This is again a streak for isolation, on this time on EMB, which stands for eosin, methylene, blue. Right, those are two different dyes that are in this agar, and those dyes allow us to distinguish organisms that are coliform. Right, these are coliform bacteria, or gram-negative enteric inside the intestines. Bacteria, based on the dyes inhibiting the gram-positive organisms, and bile salts that are in here too. This differentiates all of these organisms based on their rate of fermentation. And they all look a little bit different. So I've chosen 3 common isolates from the rectal area and given you an example of how they grow on EMB. And also all of these results. So we do something called the INVIC series to identify coliform bacteria. This is really commonly done in clinical microbiology labs with clinical stool samples, essentially, where you're trying to identify a causative agent for gastroenteritis. So with the addition of the gram stain and these INVIC series of these different biochemical testing, you should be able to determine genus and species level identification of some of the common isolates. For your laboratory activity, I want you guys to come up or identify 2. I've only given you 2 on the results page, so both of those ones I've highlighted for that MSA plate. 2 of them come up with genus and species, right? So identify them based on the gram stain and give a genus and species level characterization of them. For the throat, same thing, just choose 2 of the 3 results that I've given you and come up with genus and species level identification for these guys. And for the rectal samples, you just have to choose 1 of the 3 organisms that I provided for you and do a whole analysis of their results for all of these biochemical tests and their results on EMB to determine genus and species of that 1 isolate. For your lab notebook, I do want you to make sure you're writing down the results of all of these things and what they actually mean. So what does it mean to have a positive citrate test, for example? So to describe bacteria based on their characteristics, and then also for each of these things, at the end of this lab, for your notebook, you should have 5 genus and species identified for each of for the entire lab, 2 for nasal, 2 for throat, and 1 for rectum. Okay. There's also a normal flora worksheet associated with this one to just enhance your understanding of these things, or really test your understanding of these. So make sure you go and take a look at that one and find the due date on that one too.